発者ご紹介申し上げます。Almac Diagnostics でございます。ミスターマイケル・スローンビジネスディベロップメントを担当いたしておりまして、Almacのバイオマーカー関連サービス、診断薬およびコンパニオン診断薬の発見、開発および検査の実施ということでお話しさせていただきます。
Thank you very much. Thank you everybody for coming along this morning. It's a pleasure to be the first of the UK companies to give a presentation to you. My name is Michael Sloan. I'm the Vice President of Business Development for Almac Diagnostics, and I'm going to talk about our core business, which is the discovery, development and delivery of diagnostics and companion diagnostic tests. Before I get into that, I want to give you a little bit of context to the group of companies of which we're a part. Almac Diagnostics is part of the Almac Group, who provide services to pharmaceutical and biotech companies in both drug and diagnostic development. We have a number of divisions. We have Almac Sciences, which is a chemical and pharmaceutical development company. We have two clinical trials companies, Clinical Services, which is trial supplies, and Technologies, which is an IDRS company. And then we have Almac Pharma Services, which is a drug manufacturing company. Our European headquarters is in Craigavon in Northern Ireland, where all the divisions are based. We also have a US headquarters in Pennsylvania. And we now have an Asian headquarters in Singapore, where we have business development and project management. And we've also recently opened an office in Tokyo, where we have project management at the moment, and we're currently recruiting business development support. So we will have people on the ground with the right language skills to support our partners here. The company has been in business over 30 years and employs approximately 3,500 people. So I work for the diagnostic business unit, and we describe ourselves as a personalised medicine company focused on the discovery, development and delivery of biomarkers. What does that mean now? Because a lot of people describe themselves as that. Well, from our perspective, there are two, streams to our business model. There's our contract biomarker services business, where we very much act as a partner to pharmaceutical and biotech companies as, their biomarker or diagnostic development partner. Typically, a pharmaceutical company will not have an associated diagnostic division, and therefore that's where we can come in, acting as a service partner to help discover, develop and deliver biomarkers. And then in line with that, we also have our own internal biomarker research or product development pipeline, and I'll touch on both of these in a little bit more detail. So in terms of the service business, these are the range of services we offer. We offer biomarker discovery, where we will work and do everything from study design to profiling samples to analysing the data to help find new biomarkers. We do a lot of work in assay development, where we're refining the signatures, doing analytical and clinical validation studies to prepare and regulate those studies, those biomarkers to be utilised in both early phase trials and then on to companion diagnostics. We are running multiple trial management studies and early phase trials in our laboratories in Europe. We manage the biomarker trials, working closely with the clinical sites on behalf of our pharmaceutical partners. We offer bioinformatics consultancy because we have a core expertise there as it underpins everything we're doing. And we also then offer this retrospective trial collection. This is kind of novel, where perhaps the tumour samples were not collected in the original clinical trial, but you wish to go back and collect them. We can do that in order to collect those samples for biomarker discovery later down the line. So it's a tough enough job, but we're good at it. And then obviously laboratory services are part of what we do. And then finally, we're partnering on a number of programs in full-blown companion diagnostic development, both for kit-based assays and service-based assays. Briefly on our internal research, we also then in line have a number of our own tests, both prognostic and predictive, at different stages of commercialisation. And I'll talk in a few more examples of these again, but the test detecting recurrent stage colon cancer, a breast cancer assay, and a brain assay detecting an angiogenesis subgroup, and a prostate cancer assay. All of these developed from profiling large FFP or paraffin-embedded tissue samples, subtyping those samples, understanding the underlying biology and finding associations with those subtypes to response or prognosis, and then validating those into actual assays and biomarkers for the future. So why do people work with us? Firstly, we have the infrastructure in place to act as a service provider, as your biomarker partner in our laboratories. I'll not go through all of these, because we only have 10 minutes, but these are all of the platforms we have, both in DNA, RNA, and then we also have IHC, FISH, and ELISA capabilities. All of these selected because we believe you can actually both run and commercialize biomarkers on these platforms. And we have ensured we have a multiple range of platforms, so we're not tied to any one platform provider. We have the appropriate supply agreements in place to commercialize off these platforms, and we actually also have some of our own proprietary technology for working with paraffin-embedded samples as well. I don't have time to go through it all, but it's in the packs. In terms of quality, again, very briefly, we have all of the relevant quality standards you would expect to run both research use and clinical samples in our laboratories. Experience is another key point. And I guess it's a key factor of why we think companies should work with us, because we've truly done this. We have migrated a number of pharmaceutical companies' biomarkers into our clinical laboratory, migrating them between tissue types, platforms, laboratories, setting the thresholds, validating them and running in our labs. We've also commercialized our own assays that we've taken from discovery right through. A few examples, again at a high level. I mentioned our stage 2 colon cancer assay. Well, this is a signature of recurrence of stage 2 colon to give information to the clinician of the likelihood of response 5 years post-surgery, of which 20% of patients will respond. This was developed from profiling FFP or paraffin-embedded samples from 11 centres worldwide. We discovered a 634 gene signature, so a complex biology, which represented likelihood of recurrence. This was published in the Journal of Clinical Oncology in November 2011. We've since then entered a licensing deal with a diagnostic company in the US called Precision Therapeutics to market that exclusively in the US only. We've worked with them to perform a further validation in the CalGB, the Oncology Cooperative Group in the US clinical trial. And this validation was presented at the ASCO GI meeting in January 2014. Hence, there's a few new slides here. January was a busy month, so I updated the slide deck just before I come over. So apologies, these aren't in the pack. The test now, having validated at a hazard ratio of 2.1, which is very highly significant, is now being launched and sold in the United States. So it's a good example of how this can be done. The next example is our DDRD breast cancer assay. DDRD standing for DNA damage response deficiency. This again was discovered by profiling a large cohort of breast cancer samples in collaboration with the Mayo Clinic in the US. Again, we looked at the subtypes that came out of that research, and we discovered within those subtypes a signature response to DNA damaging agents. We then went on to validate that in breast cancer for both adjuvant and neoadjuvant, both the adjuvant and neoadjuvant setting, and published this in the JNCI in January 2014, so that's just new as well. This assay, we have now, just towards the end of last year, we licensed that to Genomic Health, who have exclusive rights to commercialize that assay globally. We're working with Genomic Health now to actually migrate that assay. This particular test is a 40 gene signature discovered on a microarray. We're migrating it to qPCR and performing further validation studies, after which the assay will be launched. What's very interesting about this approach is that you're understanding the subtypes of the disease and therefore discovering an underlying biology that you can detect using the signature of the biomarker, which you can then apply to other disease settings. And that same approach can be applied to the clinical trial samples for any drug that you may have in your pipeline for which you don't have the biomarker. And I guess that's why I throw up these examples. Other examples of our experience, we're working with most of the major global pharma and biotech companies at this stage, supporting biomarker clinical trials, multiple phase 1 and phase 2 trial enrichment studies going through our CLIA labs in Craigavon. A number of companion diagnostic development programs, including now LDT PMAs, lab developed tests. We've discovered our biomarkers both from the preclinical work where pharma companies come to us with an assay, and we then translate that into our laboratory, and then also from retrospective data sets. And we're receiving samples from over 150 sites. And finally, last few points, because we're a service company, we're flexible in our business models. We're happy to work on a fee-for-service basis for flexible IP, and we will truly partner. And And we also offer the complete package, discovery, product development, trial management, and test delivery. So I'm very pleased to be here today. We see a real opportunity. We have not worked with many Japanese pharma companies to date. I think everything that we're doing in Europe and the USA seems to be very, very relevant here. And I think there's a real opportunity for those drugs where you don't have a biomarker. We would love to talk to you about the strategies that we would perhaps put in place to help discover, develop, and commercialize a biomarker with you. And that's all I have to say. Thank you very much again.